Hello, my name is Dr. Roderick L. Roll, and today I will be talking about passive processes. Passive transport does not require energy. We will discuss a concept called diffusion. This is when molecules move from an area of higher concentration to an area of lower concentration. These molecules can move through air. They can move through liquids. The portion that we will discuss in this talk is how molecules move from an area of higher concentration to an area of lower concentration through a semipermeable membrane. This semipermeable membrane is also referred to as the cell membrane. This is the outer barrier of our cells. So we are going to further discuss how molecules move through the semipermeable membrane. The molecules we will discuss are gases, solutes, and solvents. These things will move from an area of higher concentration to an area of lower concentration. Membranes are selectively permeable. This means the membrane can allow molecules to come through, or it can restrict molecules from coming through. Movement is based on a concentration gradient. Wherever the highest concentration of the molecule is, the molecules will move in the opposite direction. These molecules are going to move through the phospholipids that make up the cell membrane, or the molecules can move through what is called a transport protein. Simple diffusion occurs when nonpolar and lipid-soluble substances diffuse directly across the phospholipid bilayer. Remember, the phospholipid bilayer is lipid-based. If we remember the amphipathic design of the phospholipid, the head is attracted to water and the tail is not. So the interior of the cell membrane is afraid of water. So these lipid-soluble molecules, these nonpolar molecules, can squeeze directly through the membrane. This is referred to as simple diffusion. Some of those molecules are oxygen, carbon dioxide, fatty acids, and some steroid hormones. This image illustrates simple diffusion. Molecules such as oxygen, carbon dioxide, and lipid-soluble molecules can just squeeze directly across the membrane. The second way we move molecules across the membrane is through facilitated diffusion. This occurs when a channel protein or a carrier protein is activated. These proteins are going to transport molecules like carbohydrates, amino acids, and ions across the membrane. The image below illustrates this concept. In the first image, you've created a pore. The channel protein is forming a hollow hole where sugars like glucose can just diffuse across based on concentration gradient. To the far right, a carrier protein is going to bind a sugar and rotate across, therefore releasing the sugar into the cytoplasm based on concentration gradient. Let's further discuss the channel protein, which forms this hollow hole. The channel protein is going to transport ions and water. You can also have a leaky channel. This leaky channel is going to always be open. One example of this would be water flowing through the membrane through a leaky channel. You do not want to restrict water flow. So the channel is always open. And then finally, we have some channel proteins that are closed. They're going to be called gated channels. So these are three types of channel proteins. This picture here shows a channel protein. It is going to allow molecules to come in and out based on concentration gradient. Again, this can be opened always, which is referred to as a leaky channel. It can be closed, therefore it would be a gated channel. And it can be a channel that moves ions. This specific channel protein is going to move water across the membrane. Remember, water can squeeze directly across the membrane in a process called simple diffusion. Water can also move across the membrane through the facilitated channel proteins. The specific channel protein that moves water is called a aquaporin. The second protein that moves molecules is a carrier protein. This is going to transport certain polar or very large molecules that cannot pass through the channels. Some examples are larger sugars like a disaccharide and some amino acids. Here we have an illustration of sucrose that is located in a higher concentration outside of the cell. It is going to bind to a carrier protein and be carried into the cell. This is also a passive process. ATP is not required. The sugar is moving based on concentration gradient. This slide summarizes all of the types of diffusion we've talked about so far. Simple diffusion, carrier-mediated facilitated diffusion, channel-mediated facilitated diffusion, and the aquaporin. Diffusion of solute across the membrane. Osmosis occurs when water diffuses across a semipermeable membrane. Remember, water can move through the aquaporin or it can squeeze directly across the membrane in a process called simple diffusion. So this is going to always occur based on concentration gradient. So there's two concentrations that we'll be looking at. The concentration of a solute versus the concentration of the solvent. The solute is the molecule, like a sugar molecule, like a salt molecule, like sodium, like potassium. Solute is the thing that is dissolved. A solvent is the thing that does the dissolving. So in this image below, we have 7 sugar molecules on one side of this semipermeable membrane. On the other side, we have 25 sugar molecules. So if the semipermeable membrane is not allowing the sugar to move, then water is going to move. Look at the image to the right and notice that the water is going to diffuse from the left going to the right to try to dilute the concentration. If the left side had 7% sugar and the right side had 25% sugar, the right side is going to be diluted so that it will be equal to the left side. So you want to have 7% sugar and 7% sugar on both sides. In order to achieve that, water has to move from the left side to the right side to dilute the concentration of sugar. That movement of water is referred to as osmosis. So in the lab, you performed an experiment. Which particle moved down the concentration gradient in experiment 6.3? So remember, in this experiment, you had a beaker that had water in it. You created this dialysis tube that also had water in it. But additionally to the water, you had a carbohydrate. You then placed iodine into the beaker. The iodine diffused across the membrane and interacted with the carbohydrate called starch. This turned starch blue-black. So therefore, which particle moved down the concentration gradient? The iodine moved from outside in the beaker to inside of the dialysis tube. Which particle was restricted from moving down the concentration gradient in experiment 6.3? Remember, you had a beaker that had water in it. The dialysis tube had water and a carbohydrate. The carbohydrate was starch. When iodine was placed outside of the dialysis tube in the beaker, it diffused into the dialysis tube. If the carbohydrate diffused out of the tube, then the water in the beaker would have also turned blue-black. So therefore, the question asks which particle was restricted from moving down its concentration gradient. That would be starch. Starch did not diffuse out of the bag. This image is showing how the membrane is permeable to both solute and water. So in our experiment 6.3, the membrane restricted the larger sugar from moving, but it did allow water and iodine to move in and out freely. So in this membrane, in this image, we're seeing that this membrane is not restricting water or the solute. Therefore, in the portion to the left, you have an unequal distribution of sugar. Eventually, sugar diffuses to the left of the membrane and water diffuses to the right of the membrane until this setup reaches equilibrium. Here is another question. The dialysis tube was permeable to which solutes in this experiment? Analyze this image and tell me which molecules moved through the semipermeable membrane. So if you look at the image to the far left, the beaker that has the dialysis tube that contains glucose and starch, Outside of that dialysis tube, you have water and Lugol solution. Lugol solution is nothing more than iodine. What you see in the middle image is that iodine diffused into the bag and stained the starch blue-black. So that means that iodine moved in to the bag, so the bag did not restrict iodine's movement. Starch is not seen outside the bag, so the bag did restrict starch from moving. Additionally, in the far right, we're using a glucose test strip. This test strip is placed in the water. Glucose is detected outside of the bag. Therefore, glucose that was inside the bag diffused out of the bag. So those are two molecules that diffused. Iodine moved in and glucose moved out. So here's the answer. Glucose moved out and Lugol solution moved in. From the diffusion experiment that you performed during lab, which does this resemble? This should look like experiment 6.3. So now we're going to look at what if the membrane restricts molecules from moving across. So that means the membrane will be impermeable. So this membrane here in this image is impermeable to solute. So the circles, the sugars, cannot move to the left for the first image. But water can move to the right. So therefore, water is going to move to dilute the concentration of that sugar. The dialysis tube was permeable to the solvent, that's water, but impermeable in allowing the solute to exit. In which experiment did you observe this phenomena? To answer that question, you have to look at experiment 6.4a. And 6.4a, you put 0% sucrose in the first bag, 10% sucrose in the second bag, and 25% sucrose in the third bag. Therefore, the end results would mean that water would diffuse into the bag to dilute the concentration of sugar, since the sugar cannot diffuse out of the bag. So the first tube that says 0% sucrose, that bag should have stayed the same weight. The second bag that has 10% sucrose, water moved in to try to dilute the sucrose molecules. Therefore, that one should have gained weight. Then finally, the last one that says 25% sucrose, more water should have diffused in to dilute the higher concentration of sucrose. Therefore, that bag should have weighed the greatest. The dialysis tube was permeable to the solvent, but impermeable in allowing the solute to enter. In which experiment did you observe this phenomenon? The answer to that question is experiment 6.4b. In this experiment, you had water in the bags. Therefore, the bags were placed in a 25% sucrose solution. Therefore, sucrose could not enter the bag. So therefore, water that was in the bag moved out of the bag to dilute the sugar concentration. Now we will discuss tonicity. Tonicity involves the solution and how that solution affects the cell volume. When referring to the solution the cell is in and how it affects the cell, we use these terms. So tonicity is related to these three terms: isotonic, hypotonic, and hypertonic. To discuss tonicity, we're going to use three cell types: an animal cell, a protista, and plants. The first one we'll look at is an animal cell. This is a red blood cell, also called a erythrocyte. An isotonic solution has the same solute concentration as the cytosol of the cell. The cell in question is a erythrocyte, a red blood cell. Inside of the red blood cell itself, you have 0.9% salt, sodium chloride. If that cell was placed in a 0.9% sodium chloride solution, you would have no effect on the cell because the outside environment is the same as the inside environment. That is referred to as isotonic. So tonicity involves the solution and how it affects the cell volume. This one is not going to affect the cell volume. How is this relevant? Whenever you go to the hospital and you've been hooked up to an IV, the solution that's in the bag is 0.9% salt. Therefore, you have no effect. If you're going to take a drug, you don't want the solution the drug is in to affect your cells. You want the actual drug to affect your cells. So this is why they use the saline drip. So again, the outside environment is 0.9%, the same as the inside environment of the red blood cell. In a hypotonic solution, the environment the cell is in has less solute than the cytosol of the cell. This means the net movement of water comes into the cell. So to illustrate that statement, let's look at the picture. The red blood cell, the erythrocyte, is in pure water. Pure water means you have 100% H2O and 0% salt. In that situation, water would flow into the cell. Why? Because remember, previously we said the inside of the red blood cell has 0.9% salt. This means it would have 99.1% water. In that case, water is going to flow into the cell to dilute the concentration of salt. In that case, the cell will swell. It will continue to swell until eventually it ruptures. This is referred to as hemolysis when the cell ruptures. Here is another way to decipher that statement. On the outside of the cell, the ECF, you have a lower concentration of the sugar or a lower concentration of the salt. So you have less solute. You would also have out there more water, a higher concentration of solvent. Inside the cell, it's called the ICF, intercellular fluid, or intercellular fluid, IN. You have more solute, more salt, and you also have less water. Let's just look at the water itself. Water is going to flow down its concentration gradient. Remember, water is not restricted. Sometimes the solute can be restricted. So let's just move the water. So the water is going to flow from the outside, the ECF, to the ICF. Therefore, the cell will swell. I also drew an image to help you understand that statement. We have more water outside than inside. We have less salt outside. So therefore, the water diffuses inside to dilute the salt. Now let's look at hypertonic. In a hypertonic solution, the environment the cell is in has more salt than the cytosol of the cell. This means the net movement of water occurs outwardly. So let's look at the last image. We have 95% water outside. Therefore, we have 5% salt outside. This would mean that there's more water inside the cell than outside the cell. So water is going to move out of the cell to dilute the high salt concentration that's outside. So the salt outside will go from 5% to maybe 4%. As more red blood cells release water, then the salt will go from 4% to 3%. As more release water, it will go from 3% to 2%. That is a hypertonic solution. So when you put salt water in an IV bag, what will occur is the water inside of your cells are going to shrivel and shrink. Why? Because they're emptying out all of the water content that's inside. To better understand the previous statement, let's look at this slide. The ECF is going to contain more salt but less water in a hypertonic solution. Inside of the cell, the intracellular fluid, you're going to have less salt but more water. If there's more water inside, the water is going to flow outwardly. It's going to flow into the ECF. Therefore, the cell will shrivel. Effects of tonicity on red blood cells. In a hypertonic solution, crenation will occur. So if the IV has 5% salt instead of 0.9% salt, you're going to have a very salty environment for the erythrocytes. What will happen to the red blood cell? The red blood cell will dump out water to try to dilute the high salt concentration. Therefore, the red blood cell will shrivel. This is called crenation. The opposite is placing the red blood cell in pure water, a hypotonic solution. This will create a process called hemolysis or hemolysis. Testing for dehydration. On average, a human cell is supposed to contain 70% water. If that level drops, the water from the ECF is going to flow into the cell. This drops the level below normal for the ECF. Dehydration in a patient occurs when their cells are in a hypotonic environment. On a hot summer day, this patient did not stay hydrated. So therefore, the ICF of their cell is supposed to be 70% water, but it drops to 50% water. Therefore, the outside of the cell, the ECF, would have more water than inside the cell. What will occur is water will move from outside to inside. Therefore, the ECF will lose its content of water. You can test the patient to see if they are dehydrated by pulling the skin on their hand. If it does not regain its turgor, then this patient is dehydrated. What can cause dehydration? Decreased fluid intake, diarrhea, diabetes, extreme weight loss, heat stroke, or vomiting. In a patient who has edema, their cells are in a hypertonic environment. This means outside of the cell has a high concentration of solute. Let's say salt. Let's say some sugar. It's high concentration. Therefore, the water inside the cell is going to leave the cell to dilute the salt or to dilute the sugar. So since more solute is outside the cell, that's where the concentration of water is going to be. The higher water is going to be out there because it is trying to dilute the salt. So in this image, a patient was stung by a bee. That venom is a solute. Therefore, outside of the cell in the ECF, you have a high concentration of this venom. The water inside the cell is going to diffuse outwardly to dilute the venom. Therefore, the area swells. This is edema. What can cause edema are all of these things listed in this slide. Now, we've talked about animal cells, specifically the erythrocyte. Now let's look at the paramecium. In lab, we saw a paramecium. The paramecium is going to use this organelle that is called the contractile vacuole. How will it use this? The paramecium is in pond water. So outside of that cell, there's more water than inside of the cell. So water is going to constantly rush in to the paramecium. The paramecium has to get rid of this excess water. So what will it use? It will use the contractile vacuole. The contractile vacuole is a pumping system that is going to pump out the excess water. If the contractile vacuole was somehow disabled, this paramecium would swell just like our erythrocytes when they are placed in pure water. On this slide, I have a YouTube video of this. When a plant cell is placed in water, the cell wall restricts membrane expansion. When a plant cell is placed in salt water, the membrane pulls away from the wall. So now we're going to look at a plant cell and how the concentration of salt affects the plant cell. So the first statement is, if we put the plant in pure water, well, plants can live in pure water. For example, this Edema grows in pure water. What happens is water is going to rush in and the cell is going to swell. But outside of the membrane for a plant, it has an outer barrier called the cell wall. The cell wall is going to resist expansion. And all it does, it adds rigidity to the plant. So this Edema placed in pure water is a good thing. But if it is placed in salt water, look at the image to the far right. This is what happens. Water is going to be expelled from the cell. It is trying to dilute the salt concentration. Therefore, the cell membrane is going to pull away from the cell wall. This process is called plasmolysis. A hypotonic environment is going to create turgor pressure in plants, while an isotonic environment is going to create limpness in a plant. So how is this relevant? When you receive your dozen of roses this Valentine's Day, make sure to put those roses in pure water. This pure water is a hypotonic environment. It is going to create turgor pressure. It's going to make your rose be firm. If you do not place it in water, it is going to be limp.